Our project is constitutive modeling of Vimentin networks under shear loading. The material that we are investigating is Vimentin networks, which is a key cytoskeleton filament that helps keep cell integrity, resilience, and tumor progression. Under shear load, Vimentin networks has string hardening behavior, which is very different from single Vimentin protein. So we want to develop a constitutive artificial neural network to model this behavior and lay the foundation for biomechanical modeling for other biopolymer networks. The stress strain data that we are using extracted from this plot in this paper. In the literature to carry out this rheological experiment, the first depolymerization used torsion pendulum to apply shear stress. Shear strain was measured 30 seconds later after the stress was imposed. Vimentin networks are anisotropic materials, so we add F4 and F5. Since it provides structural support for cells, we assume it has perfect incompressibility at 3 equal to 1. And then we use the deformation gradient of simple shear to derive the invariance and their derivatives, and calculate the shear stress, which has shear strain and shear angle parameter in it. The CAD structure that we are using has 2 hidden layers and 24 terms. The first hidden layer generates the power for 4 invariants. In the second hidden layer, we apply 3 different activation functions to it. In the first model, we use original, exponential, log, which are common choices to use in material modeling. In the second model, we replace log with soft plus, whose asymptotic behavior matches string hardening more and ensures positive stress predictions.
Tested MSE, MAE, and Hilbert laws to balance error sensitivity and robustness. We use L1 regularization to help prevent overfitting. The data was split 80% for training and 20% for testing to ensure generalization. Here's what we analyzed. We tested activation functions, loss function, and shear angles to find the best model for Vimentin mechanics. The goal was high accuracy with low complexity. Looking at different model setups, L1 activation with MSE performed the best, balancing accuracy and simplicity. Soft Plus had a similar accuracy but added complexity. Hilbert slightly reduced accuracy, while MAE overfit, capturing noise instead of real pattern. We also studied how shear angle affects accuracy. The 22.5-degree shear angle had the highest test R2 value, meaning it best captured the Vimentin shear response. This suggested an optimal balance of fiber resistance and flexibility in shear deformation. For model weight analysis, we find across all cases I5-1 squared dominated, confirming its role in strain stiffening behavior. L1 models emphasize I2-3, while soft plus models include an extra term, but it had very little effect. Here, we discuss key limitations and future improvements. The dataset was limited and might not fully capture Vimentin mechanics. Experimental constraint and biological variation also pose challenges. Future work should explain data, refine neural network architecture, and integrated tumor environments for better realism. Thank you.